right. Well, hello everybody. I'm Sabrina.
Hi, Sabrina. Nice to meet you.
Nice to meet you. Thanks for making the trip. Happy to have you here in my laboratory. So I'm the farm lab manager here. Been here for almost 5 years now. So I use this lab for quality control of all the oils that we distill downstairs. And then I also use it for research. I've published a couple papers out of this lab, which is awesome that we can do that on our farms. If you're ever curious about any of our publications that we've done, you can actually go on our website and there's a science tab and it lists all of the scientific publications that we've done at Young Living, and it has a direct link to all the papers. So I've done a couple looking at the different constituents within grand fir, conch color, and blue spruce, and looking at how they primarily exist in different parts of the tree, which is actually pretty important in our process. So it's really important that we get just the right amount of needles versus trunk materials, since certain constituents are found mostly in the needles and some are primarily found in the trunk. So that's why if we have a tree that's say like 20 years or older, we'll actually just strip the limbs off of it using a processor, and we'll just use the limb material instead of using the entire tree. In this laboratory, I run everything on this GC FID to look at the chemical profile of the oil. And then I do 3 physical tests. I do refractive index, optical rotation, and specific gravity. So each oil has its own unique raw material spec, which is a list of all the testing requirements for that particular oil. And so each one of those has requirements that it has to meet. So like the GC has a chemical profile listed below it. Each constituent has to meet a specific range. And so I use that to make sure all of our oils are meeting spec before we actually ship our oils. So I test each individual cook, which is great because I compile reports so we can look at long-term trends. So we have a harvest tab that's going to have species, age, weather information. There'll be a distillery tab that's going to have information like how long it was distilled, what the yield was for that particular cook, as well as a chemistry tab that's going to have all the different testing that I just talked about. Yeah.
Who is setting up the spec of the oil?
They do that at headquarters. There's actually an entire team that does that.
Oh.
Yeah, so they take into account a lot of different things. And since we've been in business for a really long time, obviously we have a lot of historical data that we take into account when we make these specs. So.
Thank you.
Yeah. And so then when we go to ship a barrel of oil, which typically is about, say, 5 to 7 cooks in a barrel, I'll test it a second time, and all that data is included on a certificate of analysis that they'll receive at the distribution center when they get that barrel of oil. And then the first thing they're going to do is actually quarantine that barrel, and then they're going to go through that whole raw material spec and testing before it actually gets released into production, where it's either going to be blended, which our oils go into like 30 different products of Young Living products, or if it's just going to be bottled as a single. There'll be random testing throughout production, and then once we have a finished good batch, they'll actually do another random sample pool and do one final round of testing before it's actually released for sale to all of you. So yeah, it's a lot of testing. On average, it's about 4 rounds of testing. Yeah. And our quality control lab in Utah has capabilities of like over 40 different types of instrumentation. Yeah. And then we also use third-party testing as well. Yeah, it's a lot of testing. Let's start with the GC really quick. So this is a GC FID. And like I said before, this is going to tell me the chemical profile of the oil. So we also use GC mass specs as well. So anytime I publish a paper, all of our samples, once they get received to the distribution center, they're all going to be run on a GC FID and a GC mass spec. And so the reason for that is a GC mass spec is essentially going to tell us what's there, and an FID is going to tell us how much is there. Since I already know what I'm looking at here, since I run the same oils all the time, I just use a GC FID because I just want to make sure the profile is where we want it before we ship it. And so how this thing works is it's going to pull a small amount of sample with this syringe vial out of these tiny little sample vials. This is actually this year's Melissa. Oh, yeah. Which we can smell. I'll pass these around in a second. And when I say a small amount, it's only going to pull 2 microliters, which a microliter is 1,000th of a mil. So we're talking about just a teeny, if you took a pin and drew a tiny little dot, that's like about how much sample it pulls. And so what it does is it injects it into this tiny little hole right here, which what's inside there is this glass liner. And this is what's called the inlet. So this is temperature regulated to a specific temperature to change our sample from a liquid to a gas. since this is gas chromatography, we need our sample to be in the gas state. And then these lines right here are plumbing in hydrogen gas. So the hydrogen gas is essentially just like a facilitator. It's what's going to help carry our sample through the rest of the instrumentation. So we use hydrogen because it's inert, it's happy by itself, it doesn't want to interact with the sample. Because we literally just want it to help carry it through these columns here. So these columns is where the separation occurs between all the chemical constituents that are in the oil. So this one, we can actually pass it around. What it is, it's actually a really fine capillary tubing. So this is hollow. So it's made out of fused silica. So it's glass, and it has a chemical lining on the inside. And so as our sample travels through this column, which takes a while because this column is 60 meters long, all the chemical constituents that are in the oil are interacting with that chemical lining on the inside. And so they all have a different attraction to it, which it's different types of chemical properties of the constituent, but just in general, it's just an attraction. So each one's going to have a different attraction to it. The ones that are more attracted to it are going to have more chemical interactions with that lining, whereas the ones that are not so attracted to it are just going to keep blowing through. And so what ends up happening is they all end up traveling it through at a different rate, and that's how they end up at the detector at a different time. So once a constituent gets to the detector, the first thing that happens is it's combusted. And so since our constituent was already in a gas state, we just essentially super excited and give it a charge. And so now that it has a charge and it's an ion, it can create an electrical current. And inside here are electrodes that will read the amount of electrical current that's created at that specific time. And that's what gives me a peak over here on the chromatogram, which is the end result of the analysis. So if you ever look at one of our C of A's online, which they are all available now on our website, you'll see one of these graphs. And all it is just a graph of time versus electrical current. But all these peaks are actually our constituents. So the bigger the peak, the more electrical current that was read, which just means the more of that particular constituent was present.
Hi Sabrina, have you ever encountered that the oil didn't meet the spec?
No, I haven't. Not here. Yeah. Trees are a bit easier though, because we can actually distill all year round and stay within range. Now there are other harvests, like say lavender, that are more particular, where we'll go in and just cut a small amount of plant material and do a cook in just like a 12-liter pot, like a countertop distillation unit. And then we'll run it on the GC to make sure the chemical profile is where we want it before they go and cut an entire field. So with the more finicky plants, we do have some precautionary measures that we do before we just go and start cutting things down. So, yeah. Any other questions? How are we doing on time? We got 3 minutes. Yeah, so this year's Melissa and this year's lavender that I just tested.
可以闻是吗？
From St. Mary's, yes, yes, I've already tested those.
So this farm also produces lavender and melissa and those.
No, that's from St. Mary's.
Oh,
Yeah, so we do blue spruce, grand fir. Those are our primary ones that we do. We also do evergreen essence, and then we occasionally do singles for the evergreen essence, like Western Larch, Western Red Cedar, and Ponderosa are some of the ones that we've done as single cooks for that blend.
During the cleaning process, is it before it gets here? Impurities filtering or cleaning?
I don't know what they do down. I don't think they filter downstairs. We test for impurities, but if there was impurities in there, we would just not use that oil. Once they receive it at the distribution center, that's where they're going to test for any impurities like heavy metals, pesticides, herbicides, and if those things are present, we would just not use that oil. It wouldn't be something that we try to remove. It wouldn't get used. Well, I don't know because I haven't been a part of that process, but yeah, we wouldn't be able to use it. I do have a lab down there too, but it's easier to just maintain.
Do you test every batch of the oil?
I do. I test every batch, and all that data is included on those reports that I was talking about, where I look at harvest information, distillery information, and the chemistry information. Those are great because you can look at long-term trends. But I also test every barrel that I shoot.
And how you documentation? the oil, the batch of the oil?
I keep all that in reports that we share in the quality department. And then anytime we ship a barrel, it'll be on that certificate of analysis that I talked about, that they'll receive that in an email before they get the barrel with other information like bill of lading and all that kind of stuff, shipping information.
This lab has been here ever since this farm is founded.
Well, no, because originally there was no distillery here, and they were actually trucking all the chips down to St. Mary's. So they actually had a distillation unit before we did here. They would truck it all the way down there, which is a pretty crazy drive in the winter. It's like 2.5 hours when the roads are good. So in the wintertime, Gary would drive down there. It would be pretty crazy. It would take hours. Yeah.
So the farm was first founded and then
Yes. And they built those canvas tents that they would camp in before they had anything here. So the distillery's been here now for 10 years. Right.
Time is up.
Thank you. Trying to keep on track.
Thank you.
Yes, thank